a number of spermatozoa are necessary to gain access to overcome the defenses, if you will, of the oocyte. Again, I never know when to use egg or ovum. We'll talk about a little bit about that in just a moment. But the collective action then of multiple spermatozoa will then achieve or broach the egg, and then typically only one of those spermatozoa will actually introduce the DNA. That there's a very complex process, calcium-dependent process, whereby something called polyspermy is averted. And polyspermy would simply imply multiple fertilization, in other words, more than one spermatozoa fertilizing the egg. There are mechanisms in place to prevent that from happening. If it should happen, then the entire cell will undergo what is called autophagy. It'll just destroy itself. Now, I think we've used this term before, capacitated. We've indicated that sperm are formed through a process of spermatogenesis, then spermiation, then spermiogenesis. And remember, in spermiogenesis, the spermatozoa acquire the anatomy necessary for motility, but they're not terribly motile at that point. In fact, it's ciliary action in the male reproductive tract that's largely responsible for transport. Now, by capacitation, the introduction of the spermatozoa in the female reproductive tract fully renders them motile, and in fact, you can see they are quite adept swimmers because they move all the way from the cervix to the fallopian duct. I guess we could say the distal fallopian tube or uterine tube in about 5 minutes. So that's a remarkable speed given the distance involved. Now, I've written here the egg must be fertilized within a couple days of ovulation. That's because the egg itself only survives for about 24 hours. That is to say, if it's not fertilized within, the oocyte is not fertilized within 24 hours, it tends to undergo a sort of autodestructive process. I'm not sure what to tell you about the lifespan of the spermatozoa in the reproductive tract. Just about every source has a different number. But in general, the spermatozoa can only last 3 to 4 days, I think, is really on the outside limit. And so if you see 2 days of ovulation, that's figuring into the inherent vulnerability of the oocyte and the egg cell. In a lot of the reproductive literature, though, you will sometimes read that the spermatozoa can remain viable in the female reproductive tract for 5 or 6 days. I'm not sure if that's intended to be truthful or just scary. But in any case, I think the common opinion is about 3 to 4 days. Narrow window in any case. And so, as we've indicated, in order for the oocyte to be fertilized, there must be multiple sperm attempting to gain entry into the oocyte. That precipitates something called an acrosomal reaction, where a variety of, for example, hydrolytic enzymes released by that acrosomal region of the spermatozoa are used to broach the regions you see out here, the corona radiata, which was that outer layer of granulosa cells that we looked at earlier. The zona pellucida, this sort of glycoprotein coating that acts as a sort of a defensive system. And then by the term acrosomal reaction, we're just talking about the collective activity of those hydrolytic enzymes. There are also proteases released from the acrosome of the spermatozoa, along with something like hyaluronic acid, which seems to facilitate an inflammatory type response. So there are a number of chemicals released. But as we pointed out, only one can actually merge or enter the oocyte, and a very complex sequence of events ensues involving something called a calcium wave or calcium reaction. When the sperm enters, it actually, the entire structure enters, so both the midpiece and the head enter. At that point, there's this calcium wave that is incited or initiated from the point of entry to the opposite side of the oocyte, and that can take a couple of seconds because the oocyte is so large. But when that calcium wave is complete, there is no further possibility of any other spermatozoa fertilizing the egg. And then, of course, if you remember our discussion of meiosis in the female reproductive system, we're not quite finished with meiosis at this point. Recall, once the DNA has been introduced into the cytoplasm of the oocyte, it still isn't with the nucleus. It takes another 12 to 24 hours for that to occur. But the other thing that has to happen is meiosis 2 must be completed. Remember, that involved extrusion of the second polar body. And that second polar body is because it does not contain very much cytoplasm, it too is very evanescent and disappears rather quickly. Pregnancy lasts about 40 weeks from the last menstrual period. Curiously, these numbers have changed a bit. So let me sort of warn you about the numbers that are introduced on this slide. It says implantation begins 5 to 9 days after fertilization. So what's going to happen is once that oocyte is fertilized in the uterine tube, we now have a dividing cell mass. It begins as a zygote. It begins dividing in about usually 30 hours or so. And as it's dividing, it's moving, it's passing the fallopian tube at about an 8-cell stage, something called a morula. And eventually, at about a 64-cell stage, it'll become something called a blastocyst, which has those 3 germ layers. Remember at the beginning of the course when we talked in the anatomy, we talked about the endoderm, ectoderm, and mesoderm. That's what defines the blastocyst. And that begins to appear again somewhere in the 64 to 128 cell range. But my point is, what I want to make here is that normally that dividing cell mass will reach the uterine horn, the entryway to the uterus there, in about 3 days. 3 days, but we'll be very conservative, so we'll say 3 to 6 days. Let's do that. You would expect that the dividing cell mass to reach the fundus of the uterus in 3 to 6 days. And then up there it says implantation begins 5 to 9 days. And I would say probably 4 to 9 days or 4 to 8 days. I think in general it's probably 9 days would be way out there on the extremity. And the notice down here, implantation is complete by the 11th day. Well, let's say 10th or 11th day, because again, 11 days would be out on the margin. Now, the other thing I would point out, remember we said about 3 to 6 days for entry to the uterus. Day 5, for whatever reason, seems to be a rather important day in the development of this cell mass. For example, dizygotic twinning tends to happen at about the 5th day. If there is we'll come back to that in a moment. If there's a failure of implantation, the problems usually appear at about the 5th day. So for whatever reason, there seems to be a special vulnerability at about the 5th day. When I said dizygotic twinning, what do I mean by that? What are the 2 ways, 2 general ways that we talk about twins? Yeah, they could be fraternal, in which you'd have 2 oocytes fertilized by separate spermatozoa. In fact, we'll have an even special case of that in just a moment. Or that dividing cell mass can simply split, and it can happen at about day 5 typically. And remember, that's about the time, that's in the range of the time that this dividing cell mass is entering the uterus. And there you have what would be called identical or monozygotic twins. I said monozygotic twins. You'd have the identical essentially genetically identical individuals developing simultaneously. Now, the parturition after that can be a little bit more complex, and we don't have to get into that. So implantation occurs typically again by day 10, and we have a little animation we'll look at in a second. But this dividing cell mass then implants into the uterine wall. It has to implant at just the right time and just the right place. If it doesn't implant at the right location in the uterus, that can create problems for pregnancy, or indeed it can prevent implantation altogether. There has to be the correct chemical soup, there has to be the correct timing, and so on. But at that point, once implantation has occurred, then there's sort of a union, or there's I should say, an envelopment of the maternal endometrial vascular. Remember, we saw when the uterine lining is thickening, we have this profusion of spiral arteries and spiral glands. Well, those spiral arteries and additional arteries tend to reach out and envelop the implanted blastocyst, and then those maternal vessels then provide the necessary chemicals and nutrients for growth in what's called the deciduum. So you have a little illustration here. I'm waiting for my cell. I think that's a sea urchin down there. It doesn't seem to be starting to divide. But sea urchins, people, pretty much the same. But you can see we have the embryos. We saw in the little kids taking the egg into the uterine tube. And notice the site of fertilization here, way down there from the origin of this spermatozoa. And as we've indicated, it says day 4 to 5 here, blastocyst reaches uterus. Again, we're going to get a little more conservative and say about day 3 to 6. But day 5, for whatever reason, is sort of a critical point, critical vulnerable period. Implantation, typically no later than day 9 or 10, and then you can see the sort of implantation occurring here in the wall of the endometrium. We have sound effects. Yes, this is happening in serial time. Many drive, but only one conquers. Sometimes called a pronucleus at that point. It doesn't happen that fast, and of course there's the dividing cell mass that we talked about. It gets swallowed. This process of implantation is also called nidation, and it really describes this engulfment of the blastocyst into the lining of the uterus. What we're looking at here in the old Carnegie stages, these charts and such have largely disappeared for, I presume, political reasons. But you can see the general morphology of, well, this is true for most mammals, this of course is a human, but you can see there's not much difference between us and a chicken early on. The embryo looks very similar in most mammals. But these stages, as I say, are not used as much. Little summary of the sequence of events regarding labor and delivery. I think we indicated earlier that it's not quite clear what initiates labor. The current thinking is, and this is far more complex than we need to get into in here, it's the hypothalamic pituitary adrenal axis. So probably, believe it or not, CRH is released from the um, fetal adrenal gland. So the fetal adrenal gland is a little different than the adult adrenal gland. But it's probably corticotropin releasing hormone, which you normally think about as being in the hypothalamus. It's probably the increasing concentration of CRH released from the fetal adrenal gland that is thought to be responsible for the initiation of these uterine contractions. Now, normally, to be perfectly accurate, we say that oxytocin and prostaglandins stimulate the muscle contraction, but do not initiate it. So I think that what the best I can argue is that the initiation is not well understood, but is thought to be associated with the increased levels of CRH. In any case, the uterine contractions, once initiated, intensify, largely mediated by the increasing concentrations of oxytocin or prostaglandins. Indeed, labor can be induced either with tocin, the synthetic oxytocin, or with prostaglandins. So they begin, the fetus drops to a lower position in the abdomen, normally the head's forward, if not, there can be complications. The fetus exerts some pressure on the cervix here. It says that in this slide, which is not quite accurate, the pubic symphysis and the ligaments in the pelvic outlet are softened by a hormone called relaxin. That's more true of other mammals. Relaxin doesn't seem to play much of a role in humans, at least not with respect to relaxing the pelvic outlet. The uterine contractions then force the fetus lower into the pelvis. There's dilation. There's this concerted feedback between dilation and intensification of the contractions. Oxytocin levels continue to increase. Prostaglandin levels tend to increase. And eventually the fetus progresses down the birth canal and is expelled. And shortly after that, under normal circumstances, the placenta or the afterbirth would be expelled as well. And as every mother will tell you, it goes just like this. Nothing to it. Over and done. In reality, that's rarely the case. Of course, part of the issue or part of the problem you'll have to deal with is deciding when labor should be initiated, when it should be terminated, under what circumstances is that best. In general, the current medical thinking is that the child should be allowed to develop as long as possible. You can see some of the definitions up here. I won't ask you to define these, but notice 37 and 38 weeks is early term, 39 to 40 is full term, and then you have the late term and post term after that. The idea now is you want the child to develop as fully as possible because that affords completion of the pulmonary system, the neurological system, and so on. But what other forces are at work here? Why would there be such a large proportion of early term deliveries? Which is the case. In fact, every time I talk to somebody about their childbirth, to my surprise, I hear that it was induced. So what would be the reasons for that, do you think? Okay, there could be. We talked about if the implantation does not occur in exactly the correct location, there may be very serious issues, and that would have to be addressed in one way or another. But would there be? Can you imagine, for example, again, you're the PA. The couple's coming to you and wants your advice, and of course you're going to learn about this later. But the mother may say, "I've had enough of this. This is killing me. I'm sick of this. I've been going on for week after week. I'm suffering here. Help me out."
What about when the OB has a golf thing on the weekend?
That's what I'm saying. Yeah, you've got the priorities straight. So I just want you, and again, I'm just preparing you for your clinical training, but that is part of your decision. But keep in mind that if the child is not developed fully or is not full term, usually there's some sort of consequence.
Would it have adverse effects on the mom if they let the baby go to full term? Like if the baby just goes
I would think the literature would suggest less so. Oh, are you saying? Oh, yes, yes. I see what you're saying. So you're saying, yeah, yeah.
Where it's like the baby's healthy enough, but if we let it go any further, maybe it would be worse.
Yes, you're exactly right. I was being sarcastic, but which rarely happens in here. But you're exactly right. Sometimes that is the case, and it has to be done. For example, when Ms. Ofer mentioned the placenta previa or whatever. Sometimes that is important, but I just want to alert you to the fact that when I talk, that's my experience is entirely anecdotal. It's often for convenience, and I'm just a little surprised by that, given what the obstetric literature suggests about the development of the children. In any case, there's nothing here that I would consider test-worthy. You can see that a number of hormones, you can see the levels of a variety of hormones at term here. For example, estrogen peaks at term, um, progesterone peaks at delivery. In fact, the progesterone, the estrogen level increases between conception and delivery, between implantation and delivery. I mentioned relaxin here. Relaxin, again, in humans is thought to play more of a role in preparing the uterine lining, and it seems to have much less of a role in relaxing the ligaments associated with the pelvic cavity. Here's an interesting story. A woman discovers she has two wombs. I've read several cases of this. So she has two entirely different reproductive systems. And if that weren't surprising enough, a woman with two wombs gives birth to twins. So how did that happen? Anybody want to postulate an anatomical explanation?
She could have been like two sets of twins.
Oh, I suppose. But yeah, that's yeah. What tends to happen, even in this case, is that, and this often happens with other types of twins. Implication. I want you to think about how these two are related to one another. But usually, in a situation like this, one will supplant the other. One will grow at the expense of the other. Both of these grew, and my guess is the labor had to be induced here too. There had to be cesarean, I'm sure, or cesarean, I should say so, because they would probably grow at each other's expense. But I guess what I was getting at is what is the relationship, and where did they develop anatomically? When we talk about twins.
Did they develop one in
Yeah.
each?
Yeah. And so, in theory, they could have two different fathers as well. I'm sure that didn't happen. I'm not trying to spread any rumors here. That's an aspiration of mine. Yeah. Yeah. Two different. Yeah, and that's what I was suggesting, though. But the problem with that is that the first one would almost certainly supplant the second one. That, for whatever reason, I just in my reading on this topic, because it's not uncommon for this paired anatomy to occur, but the delivery is very rare. Apparently, because one takes precedence over the other. Yeah. Because I think we mentioned earlier that you're going to learn about all a variety of problems that mothers go through in the course of pregnancy and delivery. All kinds of, for example, you're going to learn about gestational diabetes. Women convert. Not only is 40% of their cardiovascular output going to the child, but they also become diabetic. And if the pancreas can't pick up that load, the problem there, address that issue, that can present problems. There are issues of psychology and neurotransmitter balance. Yeah, I would think this would be quite taxing. Speaking of taxing, of course, so what do we mean by an ectopic pregnancy? What is ectopic? Yes, exactly right. So remember that if that zygote is not picked up, I'm sorry, if the oocyte is not picked up by the fimbrium and taken into the right location, and somehow you have an especially eager sperm, or it could happen in other ways too. It can happen in a variety of ways. You can have implantation occurring outside the normal location, outside of the uterus. So here, notice where it occurs actually in the ovary. It's very rare, but it does happen from time to time. In this case, it was successful. So, that remark. All right, any other questions there? If not, then let's begin our discussion of the cardiovascular system. This is going to be a central topic for you. Just about every aspect of your profession will involve cardiac or cardiovascular health. You can look at the, I don't like these cumulative bar graphs, but you can see this is causes of death per 100,000. Epidemiologically, the term mortality rate refers to number of deaths per 1,000. So I'm not sure what the proper term for this is. I guess it would just be mortality or something like that. Later on, you're going to learn you have to be very precise with your epidemiological terminology. But at the moment, I can't think of what the proper term is for the number of deaths per 100,000. In any case, the point I'm making, 1900 per 100,000, 137, not getting better. Right? And of course, why might that be? Any thoughts?
Obesity.
Possibly, yeah, obesity. Although, again, you're going to learn that, for example, is it better to be, if you're going into the hospital suffering a mild infarction, mild to moderate infarction, is it better to be thin, normal weight, or overweight, or slightly overweight? How are your prospects stacked up in those cases? It's better to be a little overweight. Yeah. So again, I'm just throwing out facts that are going to confound and bemuse you, because these are the things you have to deal with. With respect to obesity, I suspect you're right.
How did he arrive at this conclusion?
All right, there's a little slide here discussing the functions of the cardiovascular system. We're going to repeat a number of these over and over again, but of course the cardiovascular system consists of a central heart. The heart in our mechanical representation will be the pressure generator. It's going to generate the pressure gradient necessary for the flow of blood to occur. So over and over again, we're going to be talking about pressure and flow, pressure and flow, pressure and flow. So the cardiovascular system, through this plumbing and this circuit of plumbing, is critical for distribution of oxygen. We're going to learn that there's an important relationship, for example, between the lungs and the cardiovascular system. The capillaries will deliver the oxygenated blood to the respiring cells, the cells that are utilizing the Krebs cycle and oxidative phosphorylation to generate the ATP necessary to sustain their metabolism. The cardiovascular system is also transporting nutrients. I think we've already made the analogy between you and an automobile. When you start up your car outside in the parking lot, what are the two primary ingredients necessary for combustion? What do you need for your car to run? The very same things you need, by the way. Huh? Fuel and oxygen. Fuel and air, fuel and oxygen. You do too. You need those exact same things. In fact, I'm not recommending that you simultaneously inhale gasoline and air, but in some respects you're doing the same thing. You're taking in carbohydrates and fats, very similar chemically to the gasoline, and that products of plant decomposition, and you're combining that with oxygen. In fact, combustion and cellular respiration have a number of chemical similarities. The cardiovascular system is also collecting the metabolic waste, nitrogenous waste, carbon dioxide from the respiring cells, and then of course, depending upon the nature of the waste, bringing that waste back to the lungs for expulsion from the body, or to the kidneys for expulsion of that nitrogenous waste, and so on. We're going to find the cardiovascular system. In fact, we've talked about this when we talked about the skin. The cardiovascular system is important for regulating body temperature. Um, the cardiovascular system can either divert blood from the core to the periphery or from the periphery to the core and thereby regulate to some degree the body temperature. The cardiovascular system transports the immunological proteins and hormones, the cytokines, interferons, prostaglandins, all of these substances that are necessary to combat potential infective agents and inflammatory agents and bacteria, and fungi, and all these things. I just heard this morning on NPR that if you happen to own chickens, you shouldn't kiss them because of that risk. Because and it's even worse than that. If you read about the bird flus in Asia, in some cases how those were propagated, what seems to have initiated the disease transferring from birds to humans. Nobody's know what I'm talking about. I hesitate to bring it up because I'm a genteel individual. But apparently the farmer would be aware that the bird was sick, chicken. Don't do this either. Chicken was sick. It was stopped up, stuffy nose, stuffy beak, stuffy rostrum. And so if you are a caring farmer, bird farmer, what do you call a bird farmer? What might you do to alleviate the bird's condition? Yes. So we'll just leave it at that. And but that seems to be an important vector for communication of the bird infection. Of course, we know what a hormone is. Hormones are chemical messengers carried in the general circulation. So certainly the endocrine system depends upon the cardiovascular system for transport, conduction of these multiple hormones we've already talked about. And then there are a variety of other substances too. The liver cranes out several hundred different proteins, enzymes, and regulatory compounds that are released into the blood. There are paracrine agents that we'll talk about later that enable blood flow in your extremities to be automatically regulated without the introduction of the autonomic nervous system or the endocrine system. So there are a lot of paracrine agents and various other substances that are released into the cardiovascular system that help to regulate peripheral blood flow. Now, as you look at the section of the heart here, you might want to circle this, want to make a note, because you really need to be able to identify all the structures that you're looking at in this section. That's not our purpose at this point. We're going to go through and talk about each of these structures in a little more detail as we proceed. But what I would have you pay attention to, at least in this general, very thorough section, is that the heart, in effect, is really two pumps. It's sort of a pump on the right side and a pump on the left side, separated by this atrioventricular septum that you see here. We'll talk about the anatomy of that septum or that partition. We sometimes say that the right heart is the pulmonary side and the left heart is the systemic side. But what we mean by that is the right heart is pumping the blood into the lungs or to the lungs, and then the left heart is taking that oxygenated blood that comes from the lungs and pumping that to the rest of the body for the reasons we've already talked about. Now, as you'll notice, again, we have our two sides of the heart, and both of these in your clinical training are going to present special problems and special issues. But in addition to the bifurcation or division of the human heart into the two halves here, right and left, there's also a functional division between the basal region and sort of the apical region. Curiously, the base of the heart is at the top. And so as you'll note on both sides, in the basal region, there are atria. So this would be the right atrium, this would be the right ventricle, left atrium, and left ventricle. We'll talk about the circuit of the blood in just a moment, but in general, the blood's going to pass through, for example, the vena cava that you see there into the atrium, and then pass through this partition that separates, for example, the right atrium from the right ventricle, through this very powerful massive valve here, which for now we'll just call an atrioventricular valve, there are going to be four important valves in the heart, and what you need to keep in mind in terms of their functionality is that they are all one-way check valves that open in response to pressure. They open in response to a pressure gradient. And so, as you look at the illustration, the blood is going to flow in this direction and in that direction only, from the top to the bottom, from the base to the apex, from the atria to the ventricles. And there's some other structures we'll come back to and talk about later. For example, you can see we have these little collagenous cords in here that help to support the valve. And then attached to those cords are muscle, and we'll talk about papillary muscle too, that helps to reinforce the valvular action. So, in addition to the AV valves, we only see one of them here. There are also what are called semilunar valves that prevent blood that is expelled from the ventricles from returning to the ventricles. And what we're looking at in this particular example is the aortic valve. Now, before we continue our general anatomical overview of the heart, you'll note that the left side, this is going to be the myocardium over here. The wall of the heart is the myocardium. The myocardium on the left side of the heart is much thicker, much more robust than it is on the right side. And that is because the left side has a much heavier workload to perform. We're going to have special terms to describe that workload later. But the force of contraction must be much more powerful on the left side because the resistance to the flow of blood, to the expulsion of blood from the left ventricle is much greater. All right, so I think all of you are quite familiar with the trajectory, the flow of blood through the heart. As we've indicated, blood is going to return from the body, from the general circulation, into the vena cava. There's an SVC and an IVC, so the blood's coming in this direction and this direction. And that caval blood is going to enter the right atrium. There's the right atrium there. So the caval blood is going to enter the right atrium, and as the little arrow suggests, pass through the right AV valve into the right ventricle. From the right ventricle, it passes through what is sometimes called the pulmonary valve, this valve right here, to enter the pulmonary trunk. This is the trunk right there. And then from the trunk, the right side blood passes to the pulmonary arteries. Now, with respect to the terminology, it will almost always be the case that arteries take blood away from the heart. Veins will return blood to the heart. So the pulmonary arteries are conducting the blood away from the heart to the lungs, where the blood will be oxygenated. And then the oxygenated blood, as you can see here, will be returned to the heart by means of the pulmonary veins. The blood entering the pulmonary veins will then enter the left atrium, pass through the left AV valve that you see here, enter the left ventricle, and be very forcefully discharged via the aorta that you see here. We previously looked at the aortic valve right there. So it's going through that valve into the aorta, and then we'll talk about some of the other vessels that emerge from that structure. And the process repeats. So the blood then is going to be conducted through the body via the aorta. We'll talk about some of the vascular and mechanical aspects of the blood entering the aorta, but eventually it'll pass through all the arteries and the arterioles and the capillaries, then return to the heart by means of the venules, the veins, and eventually enter the IVC and the SVC, and the whole cycle repeats over and over. Everybody understand then the general circuit? I used to skip that, and I had a PhD student several years ago get very angry that I did, so I don't do that anymore. With respect to the discharge of blood and the pumping action of the heart, um, fairly obvious, but make sure you appreciate that blood enters from the top and leaves from the top. And when I say the top, I'm talking about the base. And the blood is going to enter the atrium, atrial region or the basal region of the heart. And then when the ventricles contract, and there are a couple of slides discussing this, the largely because of the connective tissue, both surrounding the heart and some of the connective tissue within the membrane of the pericardium and so on, the ventricles will contract in more or less a spiral fashion from apex to base. So the length of the heart is shortening, and it's also twisting, and both relative motions ensure a relatively efficient expulsion of blood. Now we'll talk about the volume of blood that's discharged from the heart, the volume that remains in the ventricles, and so on. As you look at the anterior surface of the heart here, you'll notice that most of the anterior surface of the ventricles, the apical region there, that's right ventricle. So all of this is right ventricle, and then the left ventricle is on the, it's just this portion over here, at least from the anterior perspective. And you'll also note that as we look at the heart from the anterior perspective, the apex is pointing downward and to the left. Histologically, cardiac muscle is a little different than skeletal muscle or smooth muscle. In particular, the fibers, the cells of the heart, are interlinked by what are called intercalated discs. And embedded, so this would be a cardiac myocyte. This would be cell number one. And then on the other side of this intercalated disc would be another myocyte. So that would be cell number two. But the point that I'm making here is that this Z-shaped or this squiggly shaped structure there, this intercalated disc, is embedded with gap junctions. And the gap junctions enable a depolarization in cell one to be rapidly propagated to cell two and vice versa. Those intercalated discs also contain robust cell junctions. We'll come back to desmosomes, for example. We'll come back and look at that in more detail a little later. But remember, in one of the earlier tables, we compared muscle. We used the term syncytium. What is a syncytium? Anybody remember that term? Yeah, exactly right. The indication is that there is a collection of cells, more or less acting as one. Now, we could debate, and indeed histologists do debate the extent to which cardiac muscle is a true syncytium. But in any case, the fact that, the presence of these gap junctions in the intercalated discs facilitate the unitary contraction. And we'll talk about some of the complexities that might prevent the entire heart from contracting as a unit. Now, I talked about these valves. Remember, there are going to be two atrioventricular valves. These are the large ones you see here, and two semilunar valves, and they have this general conformation in a large fibrous structure that divides the atria from the ventricles. So here we're looking at this large disc, and this disc goes by a name you already know. It's the annulus fibrosus. So what we see embedded in the annulus fibrosus, this collagenous or fibrous disc, are the two atrioventricular valves, the two openings, and the two semilunar valves. And as you look at the illustration here, what do you notice? At any given point in the cardiac cycle, something we'll talk about later, one pair is open, the other pair is closed. And when this pair opens, this pair closes. Kind of like the valves in your car.
This seems to be like a cross section. Are they all at the same level, or is this just?
No, that's when you say the same level, they're all embedded in this mechanical structure, and any displacement between the two would be immaterial to their function. Any difference in the height But yeah, they're all embedded in that same fibrous ring we talked about before. At least we used that term before. You probably remember these terms. Again, from my perspective, from a physiological perspective, I'm going to primarily use the term atrioventricular valve. But you should know that the tricuspid is on the right side. It has three flaps, three cusps, and the bicuspid is on the left side. Students traditionally remember that right tricuspid. See what I did there? And then left, mitral left, see. Um, I'm not the sort of person who would kiss a chicken because of my intellect. Um, so be sure you know these alternate names. This is more commonly what you see, and then you can see that the terms tri and bi refer to the number of flaps. So this is pretty much showing the same thing. Just make sure you appreciate that in general, this is actually, in terms of the sequence of opening and closing of these valves, this is a little bit misleading, but for our purposes and given the time that we have, I think this is a good first approximation. When you're, anybody know how your car engine works when the ignition occurs of the gas and the gas and the oxygen in the chamber there, the valves are closed, pushing the piston down. And then when that piston comes back up in a four-cycle engine, the valves have to open in order to expel the gases that were created, and then it goes down again. So it's again very, very similar. We're basically just automobile engines in so many respects. So remember the general principle here of how these valves act, and we'll take a break. The valves open, in all cases, the valves open in one direction, just like those musculovenous valves we talked about, and they open exclusively in response to a pressure difference. These valves do not have a brain. They do not have the ability to respond to anything else. It's just a pressure gradient. So let's take a break for a moment.